All right, let's go ahead and get started. So thank you everyone for joining our workshop today. I'm really excited to have Dr. Iván Godinez today to talk about Intracellular CITE-seq. Iván is the Director of Product at Proteintech Genomics, and I am Afrida Rahman Enyart. I am a Product Manager and Scientific Liaison at Proteintech, and I'll be the host for today. Before I pass it off to Iván, I just wanted to share a few company slides to introduce Proteintech to you. So Proteintech is a leading manufacturer of antibodies and immunoassays. We have a really expansive portfolio of antibodies that encompass many different research areas. We also offer immunoassays such as ELISA kits and IHC kits, and we also have other kits too, such as cell separation kits and antibody labeling kits. Within our Proteintech brand, we also have our Chromatech branded nanobody-based reagents. And nanobodies are essentially really tiny antibodies, so they're about 10 times smaller than traditional antibodies. And because they're so small, they can get really close to your protein of interest and bind with really high affinity. So they're really great for things like clean immunoprecipitation, super resolution microscopy, and protein purification. We also have our humankind branded cytokines and growth factors, and all our proteins are expressed in human HEC293 cells so that you can be confident that they have the post-translational modifications and glycosylation that you would normally see in a human protein. And then lastly, we also have our Proteintech Genomics core. So Proteintech Genomics is a subsidiary of Proteintech, and they focus on antibodies that are conjugated to oligonucleotides for single-cell RNA sequencing. And Iván will talk a little bit more about the offerings from Proteintech Genomics today. Next slide, please. So we also value that we are a company for scientists by scientists. So our award-winning products are self-manufactured, and we offer the largest in-house portfolio of antibodies for the human proteome. We also know the importance of product validation, and we use knockout-knockdown models for validation whenever possible. And as antibody experts, we do have in-depth knowledge of immunoassay applications, and we can offer outstanding technical support. So with that, I will go ahead and pass it off to Iván so that he can begin the workshop. If at any point you do have any questions for him, please do input those into the chat or the Q&A box because we will have a Q&A session at the end. So with that, I'll let you take it away, Iván. Thanks so much for being here.
Thank you, Afrida. Welcome, everybody. It's my pleasure to share with you some of the insights that we've gained about intracellular CITE-seq. But before I jump into the nitty-gritty of the technology and data, I thought I'd take a moment to make sure that we're all on the same page and start at the beginning with our oligo-conjugated antibodies. So oligo-conjugated antibodies, or DNA-conjugated antibodies, are reagents that are used to convert protein readouts, so essentially, an antibody binding to a protein into a next-gen sequencing readout. So subverting genomics to serve proteomics. So here I have a schematic of our antibody oligoconjugates, and as you can see, we conjugate a short oligonucleotide to our antibodies, approximately 50 nucleotides long. And these oligonucleotides contain three very important domains. That is our PCR handle, an antibody barcode, and a capture sequence. The antibody barcode is a unique identifier for each antibody. And now the PCR handle and the capture sequence are all platform specific. So simply what that means is if you'd like to use these antibodies with, say, a 10x genomics assay, you would have a specific PCR and capture sequence associated with that oligonucleotide. Now, through our partnership with the Proteintech Group, we have access to their wonderful antibody portfolio. As Afrida mentioned, it's the largest in-house developed portfolio for the human proteome. The goal there is to have an antibody against every protein in the human proteome. They're about 2/3 of the way through it, and we have access to all of these antibodies. So we have antibodies against 12,000 plus targets across different classes, including polys, monos, recombinant antibodies, phosphospecific and post-translational modification associated antibodies, and we have conjugated antibodies for both immunofluorescence and flow cytometry. So what does this mean for you and for Proteintech Genomics? It means that when we take an antibody and begin our screening against, say, an application like single-cell RNA-seq, that antibody has already gone through an extensive validation program across multiple applications, including Westerns, IHC, IF, flow, intracellular flow, immunoprecipitation, and neutralization assays. So when Proteintech Genomics offers an antibody, there's this rich history of validation associated with it, and we pass that experience and knowledge on to the assays we're developing with single-cell RNA-seq in this case. All right, so I will pivot now to intracellular CITE-seq and begin way at the beginning in 2017, where there was the introduction of these multiomic, particularly protein and single-cell RNA-seq assays. So the one I'm going to be discussing here is cellular indexing of transcriptomes and epitopes-seq or CITE-seq. In 2017, I have to give credit to other assays that were developed, like REAP-seq, which is RNA and protein expression seq that does essentially the same thing. So CITE-seq in this case is based on a paper that was published by Stoikius et al. At the New York Genome Center. And essentially what they did for CITE-seq is they used these oligo-conjugated antibodies. In this case, the capture sequence for their oligos was a poly A tail. Now, that was done to ensure that the oligo from the antibody or the oligo-derived tag is captured through the 10x Genomics kit, which based on this chemistry, which is the OV3, they recognize the poly A tail capture. So in this case, the cells are processed through their single-cell partitioning, which is a droplet-based encapsulation. The antibodies bound to the cells are then degraded, through the use of proteases, and the cells are lysed open. And now the mRNA molecules and the antibody oligos are captured via these beads that are included in, say, the 10x Genomics kit. And these beads contain a poly-DT chain that captures the poly-A moieties, and via size selection-based step, you isolate your cDNA libraries and your antibody libraries. So those are actually separated via size selection. Now, through this approach, it really revolutionized single-cell multiomics. And here I have some data showing, the types of visualizations that can be accomplished. So here we have a pretty common type of visualization called a t-SNE plot. Each cluster here represents a unique cell type, and each dot represents an individual cell. As you can see with these types of visualizations, we're looking at a very comprehensive view of what cell phenotypes are actually present within a given sample. And then if we look over just to the right, we can see how they visualize expression of, say, for example, CD3, the common T cell marker. And you can see that the CD4 positive T cells are lighting up for CD3 RNA and also for CD3 protein. So this is a very basic visualization that's used to communicate that we're able to detect both the RNA and the protein from the same cell. Now, when it comes to intracellular CITE-seq, that was not feasible with previous iterations of, say, for example, 10x Genomics assays. Because as you might know, if you need to access the intracellular domain of a given cell, you have to expose that cell to a detergent to enable permeabilization. However, that is only really feasible in the context of fixed cells. With prior 10x technologies, the fixation was not compatible with their chemistry because of degradation. So come in the Chromium single-cell gene expression flex, or the RNA profiling kit from 10x Genomics. This kit enables whole transcriptome profiling of fixed cells. It offers a broad sample compatibility, so it can work with fresh tissue, frozen tissue, with FFPE tissue, cell suspensions, and nuclei suspensions. Through the use of a probe-based mRNA detection assay, they've included inline multiplexing, where each mRNA target has two probes that are near exactly one base pair apart. Within one of those probes, there's an inline barcode that's sample specific. This allows you to really ramp up the scale of your experimentation and also offers a lot of flexibility in terms of the sample numbers that you're processing and the total number of cells that need to be interrogated. So for example, you can process up to 28 samples per chip and up to 1 million cells per chip. So it's a wonderful assay that really enables for us the accession of the intracellular domain and that's how we can get detection of, cytosolic proteins, secreted proteins, and transcription factors and PTMs. So the probe-based assay is as follows. You start with your fresh cells, you count them, you assess viability, and then you go through a fixation and permeabilization. Subsequently is followed up by hybridization of probes to your samples. And here we're showing that they're processing 4 different samples, and each sample has its own inline barcode, as mentioned up here. After this probe hybridization, the samples are pulled together. They undergo single-cell partitioning on the Chromium X platform, followed by ligation and extension. Library construction, again, we have our protein library separate from our gene expression libraries, which are then sequenced. They can be sequenced together or separately, and then we go in through our data analysis. So we take advantage of this workflow by adding an additional staining step prior to the flex workflow. So in this case, you would have your single cell suspension. You go through a staining protocol. Now, this staining protocol takes approximately 3 to 4 hours, depending on the options you choose for your staining. But it's encapsulated by an FC block, an optional live surface stain. So this is optional because from time to time, you'll have antibodies that don't bind very well to fixed epitopes. So in that case, particularly on the cell surface, in that case, we elect to do a live surface stain. After that, you do a 4% PFA fixation for 15 minutes. You go through a perm and block, and then you go through another round of staining, or if you're just doing this staining, that's the only round you'll be doing. And what's cool about our screening workflow is that we actually screened many of our antibodies against fixed epitopes. So what that means is, in all likelihood, you can end up just staining both your cell surface, or all your cell surface, your cytosolic, and your transcription factor antibodies all at the same time, so you don't have to break up the staining protocol. Once we've completed our staining, we can perform a few washes, which I'll discuss in just a moment. And then we enter the first step of the 10x Genomics Flex workflow, which is a longer fixation with 4% PFA for 1 hour at room temperature or overnight at 4 degrees Celsius. Once we complete this fixation, we can go on through our overnight probe hybridization, our single cell partitioning, our sequencing library prep, sequencing, and data analysis. And through this addition of a workflow, we can obtain beautiful data from the intracellular and cell surface domains of cells. So in this case, I'm showing you data for our secreted proteins. For some secreted proteins, pardon me. This is all human PBMC data, and we take human PBMCs and we take aliquots, and some aliquots we perform LPS stimulation. So this is lipopolysaccharide, which is recognized by toll-like receptor 4, typically expressed on antigen-presenting cells. Here we have PMA-stimulated cells, which, as you likely know, PMA is a very powerful mitogen and induces expression of a great number of markers. So here we have IL-1 beta in the top left, and the darker blue and the darker red indicate high expression of RNA and protein, respectively. As you can see, there's this big cluster here that's expressing both the RNA and the protein. If we look down here at the cell type UMAP, you can see that this corresponds to our macrophage population. And as expected, you can see that these macrophages are high expressors of IL-1 beta gene. And if we look at the protein, we see something very interesting. We see that, yes, for the most part, this cluster here is expressing IL-1 beta protein, but there is a very clear subset of cells that is not. And this is the type of, dynamic relationship between gene expression and protein expression that is enabled by these types of multiomic analysis. We can see into the system in a little bit more detail, and from that garner more biological insight. Now, in the middle, we're detecting the pro-inflammatory protein, the IL-17A, which is commonly expressed by Th17 cells, NK cells, and a smattering of other cell types. And here what I'm highlighting is the cleanliness of our data, i.e. the lack of background signal. You can see with IL-17, it's very highly concentrated in the T cell cluster, which is pretty expected. And then when we look at the gene expression is clustered. And when we look at the protein expression, we see an almost one-to-one relationship. And honestly, that is sort of expected because this is a highly inducible protein, and if a cell is not typically making any transcripts of IL-17A, so when it is induced, it tends to make the protein as well. And then, of course, I can't discuss any pro-inflammatory signatures without referencing interferon gamma, which is a key driver of Th1 responses. And here we can see that we can detect both the RNA and the protein at pretty high levels, very clean data as well. So in addition to secreted proteins, we can also detect transcription factors. So here we're highlighting TCF7, GATA2, and TBET. And with TCF7, it's an important transcription factor for T cells. You can see that it is expressed fairly ubiquitously, both from the protein and the RNA perspective. But one thing really caught our eye with this data, showing that in our monocyte or macrophage population, we can actually see that the gene expression levels in a subset of those cells are actually quite low. But then when you look at the protein expression levels for this transcription factor, you see that same population is actually have very high levels of that. And then interestingly, another subset of macrophages that has high gene expression levels of TCF7 actually has very little protein expression levels of that transcription factor. So again, highlighting that dynamic nature between RNA and protein expression that multipro can help elucidate for your own studies. Now, in the middle, I have a very interesting plot. It looks like there's almost nothing here. This is data for GATA2, and GATA2 is a transcription factor to help drive the differentiation of hematopoietic stem cells. Now, hematopoietic cells are pretty rare or are found in very low frequency in human PBMC fractions. So in line with that, what we're detecting here at the gene level is 2 cells expressing GATA2, and we can detect those 2 cells in the protein. Now, it looks like there's almost nothing here, and that is indeed the case. However, this plot helps us understand how sensitive both the 10x Genomics Flex and the multipro assays are, particularly when working together. So you don't lose out, on detection of these low expression proteins just by adding our workflow to the 10x workflow. And of course, we have TBET here, which is an important transcription factor for NK cells and some T cells that we're able to detect. Just again, highlighting that we can do this pretty robustly across a wide swath of biomarkers. And I think some of the most interesting data that we've generated relates to post-translational modifications, specifically phosphorylation events. So here I'm showing you data for 2 proteins, MEK1 and beta-catenin. So if we start on the right, on MEK1, we can see that the transcript for MEK1 called MAP2K1 is ubiquitously expressed. However, the phosphorylation signal is pretty much isolated to our monocyte populations with some T cell expression as well. And then we look at phosphobeta-catenin. Again, the gene for beta-catenin is ubiquitously expressed, but the phosphorylation signal is relegated to or very strongly associated with CD8 positive T cells. So very interesting data. Right, if you're interested in understanding your intracellular signaling cascades, if you want to understand how phosphorylation events and other post-translational modifications are impacting whatever system you're studying, our antibodies will enable you to do that. And in addition here, we can also detect cytosolic proteins. So these are key signaling pathway associated molecules or proteins, right? So we have things like STAT5B, SARC, SAP70. They're all associated with growth, proliferation, immune signaling. We can detect these things pretty robustly. And again, all of these things were detected, in single cocktails. So it's not like we're staining one separately. No, we added antibodies against STAT5B, against SARC, against SAP70, all in the same cocktail, and it's able to work. Now, one of the things I want to highlight with our data is that it's very clean. Obviously, whenever you're doing a genomics-based assay that's using next-gen sequencing as a readout, there should be some attention paid to the amount of background that's present. And the reason for that is that high background, while it's not going to destroy your experiments, can lead to less sequencing efficiency, where you're sequencing a lot of background signal, right? And you might be missing out on some signal associated with low expression markers or something because of that, right? So, and secondly, you're paying for it as well. So you want to ensure that your experiments have as least amount of background signaling as possible. So here at Proteintech Genomics, we spent a significant amount of resources and time developing a reagent to help mitigate the amount of background that's being detected in anytime you're doing intracellular staining. So this culminated in the development and launch of our enhanced blocking reagent, which is used for IC staining only. Where you can, if you're using this reagent, you're substantially decreasing the amount of background detected. And here I have some data showing that indeed is the case. So here we're showing human PBMCs under PMA stimulation, and we're looking at the pro-inflammatory cytokine IL-17A. You can see that if we're using standard BSA and FC block, which is something we all probably have used at one point or another, the RNA is not really impacted. You can detect, in this case, a handful of Th17 positive cells or IL-17 producing Th17 cells. You can find these same populations on the left plot highlighting protein expression. There's some obviously very dark cells here showing that those are indeed these cells, but you see a fair amount of background in all the other cell populations. When we repeat this experimentation in the presence of our enhanced blocking reagent, however, we see that the background signal is substantially reduced, leading to much cleaner data. So anytime you're doing this type of intracellular staining, particularly with our multi-pro antibodies, we do recommend using this enhanced blocking reagents. We're planning on investigating how this reagent impacts background signaling in many other applications outside of single-cell multiomics. So if this is something you're interested in, please, let us know in the chat and we can discuss that. And then, of course, at table stakes at this point is the detection of cell surface proteins. So here I'm showing you expression of the T cell marker CD3, the B cell marker CD19, and the B and T cell marker CD27. Now, what's novel about this particular set of markers is that we were able to detect these after fixation. So all of these antibodies have been validated to detect the fixed versions of these markers. So we offer access to the whole cell, right? The cell surface, the cytosol, and the nucleus. All right, so what do we do with all of this, right? So we spent a lot of time developing, conjugation capabilities, screening capabilities, and have built a really robust platform that enables us to move from unlabeled antibody to fully conjugated antibody in a very short amount of time. So we make these antibodies available to you as either singles, and I put here little QR codes for you to check out if you're interested in that. Right now, we have approximately 94 antibodies on our website, actually exactly 94 antibodies on our website. We're in the process, we're continuously uploading this. We have about 250 plus antibodies that have been validated and we'll be making it onto the website in the near future. We also offer off-the-shelf cocktails. So currently we have one cocktail called our immune profiling cocktail. It's 58 antibodies in a single reaction. They've been pre-titrated on human PBMCs and provide you very, very nice data across many important immune markers. Now, another area of specialization for us is custom services or custom cocktails and custom conjugations. So you can choose from, again, those 250 plus antibodies that we have in-house. You can also send us your own. We can conjugate them. We can create custom cocktails, validate them against cells of your choosing, and provide you a solution that's tailored to your specific questions. Now, in terms of custom conjugations, we can conjugate essentially any oligo to any antibody. What we need for there is anywhere from 0.5 to 1 milligram of unlabeled antibody, and we do this all at a very attractive price. So please, if you have any questions or need anything in terms of these oligo conjugated antibodies, feel free to chime in on the chat or let us know if there are any questions. My email address will pop up later here. All right, so now I want to segue over to intracellular CITE-seq tips. I'll start off by breaking down in a bit more detail, although not complete detail, the overall workflow. Now, I'm not going to go into a terrible amount of detail for you because it gets a little repetitive. But one of the things I want to highlight for you is that this assay really depends on a few things, and that's going to be your technique when it comes to aspirating off supernatants and the selection of the correct centrifugation systems. And I'll talk about that in just a little bit, right? But essentially the workflow is you start with your cells, you count them, you assess the viability, you spin them down, you do an FC block, spin again, you do an optional stain, spin, right? So spin, wash, or spin, stain, depending on the individual step. So this is really highlighting that there is a fine balance here that you have to consider when you're performing these types of experiments. And, these types of considerations aren't unique to, multiomic single-cell RNA-seq. These are things we've been dealing with intracellular flow cytometry, for example, for many, many years. So essentially what it comes down to is you want to make sure that you start off with as many cells as you can, and I'll talk about that in the next slide, that you aspirate and use the correct technique, and that you adequately wash your cells. Obviously, you want to end up with as many cells as possible at the end, and you want to make sure that your samples are washed thoroughly enough to minimize the amount of background that will be detected. If you ensure those two things, you will have a very successful experiment. So the tips and tricks I have are all around that. So we'll start off with the first one, and that's assessing viability. So this is a given, right? If you're already performing single-cell RNA-seq, you're aware that viability requirements are imperative, right? So we suggest that starting with 70% or greater, viability is ideal. We've seen that if you're doing these types of experiments with samples with less viability, the performance tends to be impacted. So if you have any questions or if you have any concerns around the cells you're working with, please don't hesitate to reach out to me, and I can run it through our R&D team and provide answers to you there. Our second tip is around counting cells, particularly if you're using frozen cells. So in-house at Proteintech Genomics, the way we prepare our cells is for high throughput screening, right? So what that means is we end up taking, leukopaks or large amounts of PBMCs, isolating them and aliquoting them, and subjecting different aliquots to different types of stimulations, whether it's cytokines, LPS, PMA. Post stimulations, we then take those cells and prepare them for long-term storage in liquid nitrogen. And then once we're ready to do a staining experiment, we take them out, we thaw them out. And one of the things we've noticed in our R&D is that if we let our thawed cells rest on ice for 10 to 15 minutes prior to counting, you get a better or a more accurate sense of viability of your cells. Now, tip 3 has to do with the cell number input. All of our assays are really optimized to work with a minimum input of 1 million cells. We routinely are able to get that number to work, but we have found that starting with 1 to 2 million cells works a little bit better. So if you have, extra cells, so say you're working with a cell line or human PBMCs or blood-derived cells, right, where you tend to have a little bit more cells, please start with a little bit more, and 1 to 2 million is ideal if possible. The next tip has to do with centrifugation. So as you notice in that little breakdown of the protocol, there's a lot of centrifugation that goes on. And basically, if you're performing any centrifugation, pay close attention to the number of Gs in each part of the protocol. But also, please use a swinging bucket rotor. This ensures that your pellets form nice and tight at the bottom of whatever tube you're using. And if you're using a fixed angle rotor, the pellet tends to get smeared. And then when you're going through all those aspirations and cleaning out supernatants, you tend to suck up more cells than if you were using a swinging bucket rotor. So tip 5 has to do with aspiration. So for volumes greater than 1 mil or equal to 1 mil, it's pretty convenient to use a vacuum line to aspirate the supernatant. Now, I know this is could be controversial. Some folks don't like to do it. We do this in-house, but we don't recommend that you try and aspirate as much volume as possible using the vacuum line. So essentially, we recommend that you use the vacuum line to aspirate down to about 500 microliters and then aspirate the rest with the P1000. It's important here to note that you don't have to get rid of all the supernatant to ensure successful performance of the staining. So we've routinely seen that leaving 15 to 20 microliters at the bottom of your tube next to your pellet is a-okay. And we'd rather you have a little bit of extra buffer or supernatant left over than lose that pellet. And another fun one here is using a light source. So depending on the sample that you're working with, right, fixation and the fixations we do in our protocols, right, that could lead to some cells not pelleting very well, or they are pelleting, but they're hard to see. In that case, we would recommend using a light to illuminate the pellet so that you can get a better sense of its location so that you don't accidentally suck it up with your P1000 or even your vacuum line. So in this case, we're highlighting what we use in-house. These are puck lights that are available on Amazon for a few bucks or at your local hardware store. And they really help us in illuminating the pellets so that we can see them and aspirate correctly around them. And here is one of the final tips is once you do the probe hybridization with the 10x Genomics probes, you are required to do a post-probe hyb wash. We recommend that you follow the individual wash protocol that's listed in page 94 of the 10x Genomics Flex user guide. We can give you a little bit more information there, but essentially in that last section, you do 3 post-hyb washes. We recommend that on the 3rd post-hyb wash, you spin the cells down at 2000 Gs or 2000 RCF versus the listed 850 RCF at room temperature. Now, what you do next is you remove most of the supernatant, again, leaving a small amount left behind. Use a P20 and disrupt that pellet by gently pipetting 10 times, and then add the required 0.5 ml of chilled post-hyb resuspension buffer. We do this because it helps break down the pellets and helps us avoid aggregation and helps us get better performance downstream. And then finally, the last tip is maybe just a memo in terms of sequencing parameters. I know we didn't talk very much about that. That's material for another webinar down the line. But in this case, you're definitely going to be using Cell Ranger. Depending on the version of Cell Ranger that you're using, will dictate the sequencing parameters that are required for your experimental run. So if you're using Cell Ranger 8 or above, read 1 will be 28 cycles, read 2 will be 90, and 10 for each of the index reads. Whereas if you're using Cell Ranger 7.2, read 1 will be 48 cycles, and read 2 will be 50 cycles. So at this point, I'd like to wrap up the main component of the webinar and open up for our Q&A session.
Great. Thank you so much, Iván. That was a wonderful talk. We have quite a few questions to get through, so we'll go ahead and kind of dive into the Q&A session. I know Kit has answered a few of the questions in the chat, and Kit is the CEO of Proteintech Genomics, so he definitely knows what he's talking about. So if you got a response from him, you definitely got the right answer. So we'll start with the first question here, which was a question that actually came up during this morning session as well, which is, do you have any anti-mouse antibodies or only anti-human antibodies?
Yeah, so the big focus of Proteintech Genomics has been around the human proteome. But most of our antibodies that we develop, we ensure target conserved epitopes. So if you look at Proteintech's portfolio, you'll see that a large or a significant portion of their antibodies are actually cross-reactive across multiple species, including mouse, rat, cow, dog, chicken, right? So there's been a lot of cross-reactivity validation performed there. So while most of our antibodies are geared more towards human proteins, they are geared toward or target conserved epitopes. So in terms of the mouse work we've done in-house at Proteintech Genomics, that is something that is on our roadmap. We will begin screening our antibodies against mouse samples, so splenocytes and other cells derived from different tissues. And we hope to complete that either by the end of this year or early next year. So it is coming. You know, the single cell world is split pretty evenly between mouse and human cells. So we want to make sure that we serve both of those communities adequately.
Yeah, and if you sign up for the Proteintech newsletter, we do, send out emails anytime we have a new product launch. So if you are interested in that, you can always sign up for our newsletter to find out more about kind of upcoming products. Our next question is, do you need to fix and permeabilize the sample prior to adding your intracellular antibodies?
Yes. Yeah. So that fixation, that initial 15-minute fixation provides the cell enough structural integrity so that when we subject it to detergent, it doesn't fall apart, right? So you do the fix, makes the cell into Peter Smybert says they turn into little rocks. And then you do a little soap, and that permeabilizes them a bit, and that's actually what enables the access to the intracellular domain. So without those two steps, yeah, you won't be able to get intracellular staining.
Great, thanks, Vaughn. Um, our next question is asking for the custom option. Is there a option to do a custom protein, or is it just a panel?
I think I need a little bit extra information. You mean like a custom antibody against a single protein? That is something we can definitely do, and we do offer as a service. But if you mean like if you want to give us a peptide and have us conjugate something against that, I think we can do that, but I'd have to double check with our conjugation team to ensure that is indeed possible. But if you can provide a little bit more details on exactly what you would like us to think about, that would be helpful.
Yep. You can feel free to clarify that in the chat and I can address it, or you can also just email Iván if you want to chat about it more. Our next question is your method also applicable to single nucleus RNA-seq?
Yeah, so yeah, single nuclear RNA-seq, right, is a pretty big area. We have partners that have performed work with single nuclear RNA-seq with our cocktails, and that is something that we are focused in developing as well in-house, but we're not quite ready to share our data. So the short answer is yes, we will be releasing that our any single nuclei associated antibodies pretty soon. So stay tuned.
Great. Our next question says, is there a maximum number of antibodies for intracellular CITE-seq?
The short answer is not really. We routinely do experiments in the lab of several hundred antibodies at a time, spanning, intracellular, cell surface, nuclear. There are some practical things to consider though, is if you go really high, like say 500, 600, 700 antibodies, you're introducing a ton of protein into a given cell, right? So the amount of blocking and stuff needs to be optimized for those larger types of experiments. But in terms of just theoretical overlap, we're not really concerned about spectral overlap or anything like that. So there's really an unlimited amount of plex that's available for these types of experiments, simply because it comes down to the different number of nucleate barcodes associated with each antibody. So each one is 15 nucleotides long, or each barcode is 15 nucleotides long, and there's 4 possible positions in each one of those positions. So you can do the math and see like there's a lot of space available in terms of plexity.
So the limiting factor is how much data do you want to analyze?
Yeah, that's the other question. With sequencing, yeah. So I mean, it's definitely possible. We do it in-house, and if that's something you want to do in terms of a very large cocktail, just let us know. We have tons of experience in this space.
Right. The next question is asking, so if I input 1 million cells, what is the expected cell recovery rate?
Yeah, that's a great question, right? And it really varies on like the sample type, how many antibodies, your technique and stuff. But what we see in-house is that ranges from 40 to 70% recovery.
Great. Our next question is saying, what number of cells do you typically recommend starting with and sequencing for the Flex kit? And viability cutoff does not apply for this kit?
I'm not sure about the viability cutoff question there, but the number of cells that you input, I think you can get away with like 8,000 cells at the end that you load onto your gem. So there's quite a bit of flexibility in the kit, right? So if you're starting with 1 million cells and you end up with 200,000 or something per sample, that's more than enough to run on the Flex kit. Yeah, so viability, I think what you're saying is, since you are working with fixed cells, that viability is not a concern. And I think while there's an element of truth to that, something you have to consider is if your cells prior to fixation, right, that longer term fixation, are already dead and there's a lot of kind of gunk and junk around there and stuff. If you fix those cells, yes, you will be able to put them into the kit, but what you get out of it at the end of the day is going to be fewer usable cells, and it's just not going to perform well. So in that sense, yeah, viability, it's less of a concern, but you still want to make sure that when you do fixation, that you start with, a good viability.
Great, thanks, Iván. We have time for a few more questions. So the next question is it possible to use multiplexed barcodes of fixed RNA profiling together?
Multiplex. Yeah, so I think I understand your question. But so the way the kits work is you can have up to 16 sets of whole transcriptome pooled barcodes, right? And so that means you have 16 samples you can multiplex together. So you can pool them together, and that inline barcoding that I mentioned will enable you to demultiplex individual samples after they've been pulled together.
Right. Someone's asking if we provide kits so that they can conjugate their antibodies themselves.
So that's something. Yeah, we are interested in providing. So our parent company, Proteintech, offers a wonderful reagent called Flexible that enables benchtop 10-minute protocol for essentially conjugating fluorophores to your antibodies based on species and isotype. We are working on developing reagents that will enable you to do that with oligonucleotides. Again, just keep tuned, and we'll be providing information when we're ready to launch that.
Perfect, thanks. So there's a question saying, do I need to design custom probes for readout to read out these proteins in the Flex method, or nucleotide
No, no,
conjugated on the proteins can be directly
yeah.
captured by the 10x gel base.
Yeah, no, you don't have to design any probes. So there is an accessory kit you would have to buy with the Flex to ensure that you're getting your feature barcodes read out. But other than that, it's all included. There are oligos that capture the antibodies in that kit if you get that accessory kit. And so you don't have to do any probe design or anything like that.
All right, great. There is a question about the TCF7 and IL data that you showed. So they were curious if there was any other validation steps that were taken to ensure that they weren't artifacts, mainly because high RNA but low protein seems to make sense, but low RNA and high protein is less obvious. So do you have any
Yeah. So
ideas on that?
that's a really good question, right? And we've done experiments where we leave out things like the protein transport inhibitors, right, to see if there's any artifacts that are being generated by that. What we've seen is that no, I mean, we don't think these are artifacts based on repeat experimentation and detection, but also even within those same data sets, right? We do see that some cells the staining is working robustly in those populations. So that leads us to believe that whatever we're seeing there is happening, is real, and is not an artifact. Now, whether we've gone through and tried to address that mechanistically and done subsequent studies trying to look at those specific situations, the short answer is no, and we leave that to the greater scientific community if that's something you're interested in. But obviously that the nature of protein gene expression dynamics is a big question. And it's an area that we feel will be very fruitful in terms of providing insight to researchers on whatever systems that they're studying.
Great, thanks, Iván. Here is kind of a little bit more of a specific question. If you're aware of any instance where this assay has been used for mapping the immunogenic epitopes on a therapeutic protein that are bound by anti-drug antibodies and/or neutralizing antibodies. So this would be essentially B-cell epitopes on the surface of a soluble protein.
Yeah, so I think that's not something that we've attempted, but if that's something you want to work on with us, let us know, and then we'll be happy to collaborate with you.
Great, thanks. And then we just had a question that came in asking, are you able to perform cell/nuclei hashing with intracellular CITE-seq?
Great question, right? So for those of you that don't know, hashing is a way of increasing multiplexing capabilities on top of like the Flex inline barcoding system that they've developed, or 10x has developed. So the short answer is yes. You can use cell surface epitopes to help drive cell hashing. Both, as I mentioned earlier, we do screening against fixed epitopes. So we're looking right now to develop and release a cell hashing kit that'll work with both fixed and live cells. So yes, it's something that you can do. We've done it in-house. We're still kind of new in this space in terms of we've been around for a few years, and as individuals, we have lots of experience. But as a company, we launched our first product in November, and we're doing really tons of development work right now. And we hope to launch a lot of kits along cell hashing, nuclei hashing, and other relevant assays sometime this year. So we're working on it for sure.
Thanks so much, Iván. So this has been really great. It's always really great to hear you talk. You're an excellent speaker, and every time I feel like I learn something new from you. And thank you everyone who attended. If you did like this workshop and want to know about any of our upcoming events, please do follow us on our social media because we do post about our events there. I do just have a couple more slides to share some more information with you. So I just wanted to let you know that, if you're interested in learning more about genomics and next generation applications, such as multiomics, scRNA-seq, and spatial transcriptomics, you can check out our Genomics Learning Center that we have on our website. So you can just scan the QR code and it should take you directly there. It's got a bunch of great resources available for you. If you're interested in learning more about MultiPro, you can also read through some of our general FAQs if we haven't covered them during this webinar. And you can also, directly reach out to Iván as well at ivan@ptglab.com if you have more specific questions for him. And then I just wanted to end with this. So as an attendee of today's webinar, you will receive a follow-up email that will include the recording of this webinar. You'll also receive a separate email that will give you a certificate of attendance. And then you'll also have access to use the following promo codes. So the first one is 50% off MultiPro Human Immune Profiling Cocktail and Enhanced Blocking Reagent. And the second one is buy 3, get 2 free single antibody conjugates. So we will be including those codes in the follow-up email as well. And please do allow 1 week for this email to be sent. So with that, I want to just thank Iván once again for his time and for sharing everything about Intracellular CITE-seq with us. And of course, thank you to everyone who attended. So with that, I hope everyone has a great rest of your day, and I'll go ahead and end the webinar. Take care.